So up next, we have Professor James Walters, who's Professor of Psychiatry and Director of the MRC Center for Neuropsychiatric Genetics at Cardiff University. And he's going to be giving updates from the Schizophrenia Working Group of the PGC.
James, you're muted. That's better.
Yeah, can
you hear me now?
Yeah, now we can hear you.
Okay, thanks. So my first sentence was, can everyone hear me? And obviously the answer was no. Thank you, Andrew and Talia, for chairing the session. I'm very pleased to speak on behalf of the schizophrenia group of the PGC. I'd like also to thank Topher and Jerome for putting together such a great session and conference. And it seems to work very well. So well done for all the background work that's gone on there. I thought I'd today, really. We've been trying as hard as we possibly can to get the PGC3 paper over the line. And so whilst I can talk if people are interested in the various initiatives within the group, I thought really to concentrate on the results of this paper and how those are developing, people would want to hear. So that's what I'm going to focus on. Before I do that, just as a general overview about schizophrenia, really. So this is a picture drawn by Henry Coburn, who's the son of a very well-known UK journalist who developed schizophrenia, and I think depicts crucial elements of the disorder. And so schizophrenia is characterized by groups of symptoms: positive psychotic symptoms, negative symptoms, which are more related to emotional and social withdrawal, and then cognitive impairment. But it's the psychosis symptoms that are really the classic symptoms, and they're typically of this persecutory paranoid nature that picture depicts. The other thing to note is that the condition is associated with marked economic disadvantages, poor housing, often people with severe forms of the disorder are in long-term care or homeless. And we know from modern epidemiological approaches that urbanicity and also migration are associated with increased risk. Certainly in the UK and US, African ancestry is also associated with increased risk, probably related to the socioeconomic risk that I've described above. And the other thing to note is that for we think probably self-pharmacological reasons, the rates of smoking are very high in those with schizophrenia, as are the rates of physical ill health and obesity. This lead into a decreased life expectancy of between 15 and 20 years. I highlight these factors to make a point, really, and that is that it's no coincidence that these are also all factors that could be associated with susceptibility to COVID, but also poor outcome in COVID. And I think we've heard about this during the meeting, but it bears pointing out again that these are groups of people, and this isn't exclusive to schizophrenia, it applies to people with severe disorders across the piece, really, particularly perhaps people with learning disabilities, but people that are going to be suffering more than anyone at the moment because of these features. And they're not people, unfortunately, that are going to engage with social media a lot of the time. And so the sooner we can get back to face-to-face contact and support for these people, all the better. But it also, I think, emphasizes the importance of the research that the PGC and everyone are doing, because we're talking about groups that are susceptible in so many ways to physical ill health. Okay, so if we made that point, I'm going to go on to say what we've been doing to try and remedy that situation as far as genomics and moving towards therapeutic targets are concerned. And so what I'm going to do is outline the results of the most recent PGC3 study. I'm going to talk a bit about polygenic risk and how that develops with increasing sample size, and how statistical prediction and clinical prediction emerge from that, or lack thereof. I'll then talk about some of the gene set analyses that we've been able to do to try and get traction on biology. And then I'll talk about a particular aspect of the paper that I think is moving things forward, which is about statistical fine mapping and how that can help us with prioritizing genes for functional and perhaps therapeutic follow-up. And then finally talk about the results of common genetic variation in tandem with those of rare genetic variation. The schema is a consortium that many members of the PGC schizophrenia group are involved with, which is relating to whole exome sequencing analysis. Okay, so firstly, this is our phase 2. So this is the Nature 2014 paper, and these are the results from the more recent iteration. And as you can see there, it's greener and it's taller. There are more genome-wide significant hits, and those that are there are more significantly implicated. And what we've done for this GWAS is bring together data now on over 70,000 cases and roughly 100,000 controls. 80% of those are of European ancestry, but there's a meta-analysis with those of East Asian ancestry who constitute 20% of this sample. We're still not doing well enough in that trans-ancestry respect, but there's movement there, certainly. And that gives us a SNP-based heritability of 24%. And with these findings together with meta-analytic data from DECODE, then there's 329 independent associations, and those collect together for 265 genome-wide significant autosomal loci and 4 loci on the X chromosome. So in increasing the sample size by 80%, we get over 150% increase in the number of genome-wide significant hits. And I think it shows that with an increase in sample size, you're getting new findings. I think that will hopefully be a recurrent theme throughout the next 10 minutes or so. This is a really quick slide. So this is an issue that refuses to die, and we thought we'd address this in the best way that we can. So are there genetic differences between those who are male and female? So there is a slight male preponderance for schizophrenia, but as far as we could ascertain, there are no significant genetic differences. So there was a high genetic correlation when you run a male versus female schizophrenia GWAS. There were no significant findings. And so everything we were able to ascertain suggested that the common variant signal for schizophrenia was consistent between males and females. I'm sure it won't put this to bed, but that was the intention. Okay, and then moving on to the polygenic work. So what previous versions of the paper have done is taken as the polygenic target population the molecular genetics of schizophrenia study. And here we've done that with the sequential versions of the GWAS training set. So back to 2007, and then the 3 iterations of the PGC study. And you can see this uplift of liability explained for polygenic scores. So this is from the 70 or 69 samples and then targeting the MGS. And that gives a liability, heritability on the liability scale of 10.4%, and the genome-wide significant hits alone explain 2.7% on the liability scale. And so you can see that there's an incremental but not a massive uplift between PGC 2 and 3 as far as all the SNPs are concerned, but this large increase in the liability for the genome-wide significant hits of over double the rate, which I think is interesting and tells us we're on the right track. What we then went on to do was, so people may know of this paper, which was focused for coronary artery disease, the Kira paper from 2018. And what the argument they mounted was that polygenic risk, if you take the top 10% of the distribution of polygenic risk and compare the people's risk in that group compared to the remaining 90% of the polygenic distribution, then this is equivalent to monogenic conditions was the case they made in that. And so we thought we'd compare our results to those results. And when we do that, essentially the results in schizophrenia are comparable, but actually stronger than those in coronary artery disease. So if we take the top 10% of the distribution compared to everyone else, then your chances of having schizophrenia as opposed to being a control in that top 10% are about 4.46 odds. And then for the top 1%, that goes up further. However, I think this is sobering given the prevalence and incidence of schizophrenia, that in no way I think at the moment extends to any useful clinical prediction, certainly at a population level. So I think if polygenic scoring is to be useful in that respect, then what we need to do is target prediction in a population where the prevalence of the condition or an outcome is far greater than the 1% population prevalence. What we also wanted to do was compare results across ancestries in this sense. And so these are the results comparing European and Asian ancestry. So the top yellow panel here is European and the bottom red panel is Asian. And then we've looked at, in these columns, the trans ancestry training set, so European and Asian combined, and their prediction at these levels in both ancestries. And then an ancestry specific training set polygenic score predicting, so European on European prediction and Asian on Asian prediction. And the message here really is that the trans ancestry training set outperforms the ancestry specific training set for both European and Asian ancestries. So there's benefits to having a trans ancestry PRS enrichment, and that signifies high genetic correlation between ancestries, but it's still beholden on us, and for any number of reasons, to develop further ancestry specific prediction if polygenic scores are going to be useful, but also for wider reasons in both Asian but also wider ancestries. And that's something we, along with many other groups, are actively trying to do. Just to touch on gene sets, and so trying to focus in on biology at a very broad level. So this is the generic gene ontology analyses, and the only point to make from this slide really is that those sets that emerge are are exclusively, even at trend level, restricted to central nervous system gene sets, and no other gene sets are implicated. And then the other broad point for this generic gene set Go enrichment analysis concerned is that it's the usual suspects really, so synapse and synaptic processes as well as ion channels implicated in this new gene set analysis. We wanted then to go further and look, and we were able to work with people that have done further annotation of synaptic gene sets. So this is the SINGO consortium, Hagen and Hugh Smith, who've been very helpful in this respect. What I've put here is, so these gene sets are split into biological processes and cellular components, and PGC2 gave us this picture, whereas you can see from PGC3 we're getting more granularity as far as the processes and cellular components are concerned. And these really highlight specifically postsynaptic protein complexes and signaling, and this level of functional specialization at postsynaptic and some transsynaptic processes, but particularly postsynaptic. And the signal seems to derive from there rather than it does other synaptic gene sets. The other thing we wanted to do was to try and start to address this issue, which is that, okay, we've got association across a locus, and you can see the zoom plot typically here, but this tells us little about the genes that are implicated. And so to move from this general association down to hopefully lists of more credible SNPs, then we used statistical fine mapping and also further annotation methods that I'm not going to talk about in detail, but I want to go into the fine map results because I think that approach hopefully will be informative. And so what we did was statistically fine map together with the people that designed fine map and developing the LD reference within the data set itself, which I think has particular strengths, and then determined a set of credibly causal SNPs. And at each locus, FineMap will tell you the most likely model for the number of causal SNPs at that locus. And actually, when we did that, we saw that, so this depicts the expected number of causal SNPs, this number K, as far as all the genome-wide significant loci are concerned. And so the majority of loci, there's a single causal variant predicted from FineMap, and just under 90% of loci have a number of predicted causal variants of under 3. So we've decided subsequently to focus on these. It's pleasing that this approach is also highly consistent with an independent approach of transancestry mapping, and so they both seem to be highlighting sets of credible SNPs that are candidates to be taken forward. And so using that, how did we then prioritize genes? Well, just using the FindMap data in itself, then we took single non-synonymous variants for the posterior. So the FindMap gives you a posterior, Bayesian posterior probability for each SNP. And it's those collectively those SNPs that constitute the 95% posterior probability group that constitute the FindMap credible set. But we took those non-synonymous SNPs with a posterior above 0.1, UTRs about 0.1, and then took those genes, prioritized genes on the basis that all the FindMap SNPs that in that 95% credible set lie within the boundaries of that gene. And we looked to validate that, and we did that by looking to see if those prioritized genes have an enrichment for genomic features we know are enriched for schizophrenia. So loss of function intolerance and brain expression. And indeed, the prioritized genes had a greater loss of function intolerance metric and were more likely to be brain expressed than other genes in the loci, which was pleasing for this prioritization approach. And that gives us a set of genes across each of those, some of which people will have seen before, but some of which are novel. So this is a well-known variant in the manganese transporter, a missense variant, but actually the posterior probability for that is now up to 0.99. I highlight IRF3 as well. So this is involved in regulation of interferon and missense mutations within this gene, particularly interferon alpha and beta. Missense variants within this gene are associated with herpes encephalitis, which is one of the major organic differential diagnoses of schizophrenia, which I think is interesting. Furin, I think we might have heard of elsewhere in this meeting, and there are also from the single gene analysis interest in transcription factors, but also neurodevelopmental and glutamate specifically genes. We then went on to further prioritize genes based on SMR plus other methods, and I think I'm not going to go into those in any detail, but just to say that there was this bilateral approach to prioritizing genes, and in total we prioritized 108 unique genes from these various approaches. The other thing I want to touch upon today, because I think it's really nice, is the analysis that we did with Schema, so the Schizophrenia Exome meta-analysis consortium led by Mark Daly and Ben Neal, which, as I said, lots of PGC people are part of. And this is looking at ultra rare variant, single nucleotide variant association. And if we look at the prioritized genes that we have, and then look at the schema associated genes, so schema identified 10 genes at a genome-wide significant level and 35 genes at an FDR less than 0.1 level, then what we see is some limited, but you know, of the prioritized genes that we identify from FindMap, there's overlap with those schema genes, particularly at GRIN2A and SP4, which is a transcription factor. And then we looked more broadly to see whether schema identified genes show enrichment in the common variant signal. And because in the rare variant field, schizophrenia, even with schema, lags behind in power, autism, and developmental data sets, we wanted to look at those as well. And we did that using MAGMA, controlling for brain enrichment, gene length, etc. So this slide describes those. So this is the result for the schema genome-wide significant genes, 10 of those. And if we look in the PGC3 GWAS data, then we see, you know, minimal enrichment, but nonetheless convergent signal between the GWAS and rare variant signal. And this strengthens as the number of genes hit and the threshold for the cutoff in schema is lowered. Which I think is really pleasing. If we then look at autism spectrum disorder and the most recent D trio de novo variants, then they show marked enrichment as gene sets for MAGMA analysis for schizophrenia GWAS. So we're seeing convergence for schizophrenia in rare variant studies, and hopefully with the increasing power of subsequent data sets for rare variant discovery, that will increase. But at the moment, very strong enrichment for other neurodevelopmental disorders. So rare variation and common variation converging, which I think is really important and will lead to hopefully therapeutic development. So to summarize then, what I've said is that certainly for schizophrenia, larger GWAS continue to pay dividends, and we're finding, particularly when you're trying to gain biological insight, it's not mechanistic yet, but biological insight is ramped up with sample size and increased inheritability, albeit that minimal. The interpretation with new functional annotation methods is becoming more tractable, I think, and we certainly think that statistical fine mapping can help focus the search space in that respect. And synaptic annotations seem to be important for schizophrenia particularly, but it's likely to be the case for other neuropsychiatric disorders. PRS prediction is improving, but I really think we've got to be cautious about how that can be clinically applied, and we need verification of that prediction before we start to talk about this being clinically useful, because of the risks of doing that to the wider field, perhaps. And then, as I ended on, there's this convergence between rare and common variation, which I think is hopefully going to lead us forward. So that's pretty crude at the moment at biological level with limited overlap, but certainly at a genic level, at a broader gene set level, there's quite strong convergence. And then finally, it only leaves me to thank particularly Mick O'Donovan, my co-chair, who's really led a lot of this work, and to Vasa and Antonio, who've done tremendous amount of work, as has Stefan Ripke, and to the wider group, and lots of collaborators who aren't in the schizophrenia group themselves, but have really lent their stat gen, functional annotation, expertise for these sets of analyses. And hopefully the paper, I've said this before, to my disappointment, it's not out yet, but we really hope that it will be in a short amount of time. And thanks for your attention. I'm happy to take questions now.
Thanks very much, James. That was a really informative talk and really useful for everyone to hear how the schizophrenia field is progressing.